So, hello and namaste to all of you. This is Name-Shalu Singh from Department of Physiotherapy and Diagnostics from Jayoti Vidyapeeth Women's University. So, in past sections, I've told you about lots and lots of parasites. So, I'll be continuing with those again. So, for today, I'll start with Tricus Tricuria. So it is also known as whipworm, and it looks like a tiny whip with a handle and lash. And now these are 3 to 5 centimeter in length and white color. And now next, the eggs can be 25 to 50 micrometer sized and brown colored and double shelled. Outer one is bile stained, and these are thick shelled and barrel shaped with mucus plug on both the poles. Single ovum can be seen, and it floats in saturated solution of your common salt. Nextly, the next worm can be a strongyloid stercoralis, which is adult worm, and the females are readily discovered than males, and it has 2.5 to 40 uh, into 40 to 50 micrometer in size in case of females, and posterior extremity is pointed, and in males are shorter and broader than females. uh, eggs Nextly, eggs are conspicuous with body in a single line and 55 to 30 micrometer, thin shelled, transparent oval, and they contain larvae ready to hatch, and the larva, not the egg, are found in stool. Nextly, there can be rapidiform larva and filiform larva. So rapidiform larva develops directly from the gravid females, and their short mouth double a bulb esophagus, whereas a filariform larva has longer slender shape with short mouth and cylindrical esophagus. Now, further, helminths, either that float or that do not float, are in your saturated solution of saline. So various helminth eggs that float can be that of Duodenale, of your N. americans, E. vermicularis, H. nana, E. lumbricoides, T. trichuria, and H. diminuta. And eggs that do not float can be of A. lumbricoides, T. solium, T. saginata, trematodes, F. busci, and F. hepatica, and C. sinensis. Now, various artifacts can be seen in your stool. So what basically artifacts are here, that artifacts or other things, they are living or artificial and present in stool that are not parasites and could mislead for the to your laboratory workers. So artifacts are generally not mistaken for cysts actually. So these artifacts, they are sometimes mistaken for your worms and for your parasites and for your mostly for your casts and crystals, or for your um, eggs present in your stool. So now stool culture is done. So stool culture is used to detect the presence of disease causing pathogenic bacteria, and it is helpful in diagnosis of infection of digestive system, and is used in conjunction with stool test, and the reference range of stool culture is always negative. Now, some common bacteria for stool of cultures for culture is Campylobacter species, Salmonella species, Shigella species, Yersinia species, Vibrio species, and Vibrio species are mainly found in people having travel history, and some bacteria, they cause illness by producing toxins, and they can be detected by your PCR antigen test, which are done for your school examination. Next, your E. coli and your Clostridium difficile. Nextly, collection specimen is collected via rectal swab in infants. Sterile collection container is not required, so no detergent or preservative should be present in the container. And specimen should be immediately transported to the laboratory. And if transport is delayed by longer 2 hours, transport of media like a carry blair is recommended. So samples must be sent in sealed depot container marked with biohazard sticker. Now, the media that I commonly use is your McConkey agar for Salmonella and Shigella species, eosin methylene blue agar, triple sugar TSI, which is a differential for Salmonella and Shigella, your separate agar, Hecaton, enteric agar, and your selenite broth. Uh, Nextly, this was all about the culture, and your culture is done to have a detailed examination. So first you have to read you can also, if you're curious, you can also read about these types of agars and the what are all are, how are they detected. So this was all for today. Until then, until next class, take care and goodbye.